Good morning, everyone. Thank you, Colleen, for your introduction. And today I'll just give an update with our work on the text mining projects, which we have set up in collaboration with the University of Illinois in Urbana-Champaign with Professor Jiawei Han's team. And I'll show you our data and how we are so far. So the Medline database is using PubMed as a search engine, boosts over now 2.2 million cardiovascular related articles. And they have been piling up, accumulated since 1809 to present time. And it's estimated that about every 2.7 minutes, there's a new publication. So it's clear that there's an enormous growth of textual data, especially in the past decades, to enormous amounts. And these are basically still unmatched by suitable analytical tools. And with these much data available online, we can analyze the data to discover new insights. For instance, from many omics studies that are now available or clinical trials. And also now clinical case reports are an important source of information and that have been accumulated online. And then there's also in the hospital the accumulation of electronic health records that have become massive and unstructured. So all these sources of data, they form new challenges to conduct new investigations and to actually also facilitate precision medicine. So in view of all these new challenges in data science and in the realm of unstructured textual data, what we do now is we aim to address these challenges by developing new text mining and also machine learning technologies, so we can dissect all these enormous amounts and volumes of textual data, in our case on cardiovascular disease. And basically what we would like to do is identify patterns in data sets and recognize the hidden mechanistic insights. So here are the aims of the study. What we would like to do is we like to conduct a phrase mining and machine learning algorithm or angle on vast amounts of textual data. In our case, we have about 1.1 million abstracts that we've been analyzing, and we try to look at the relevance of 8,325 heart-related proteins that we identified in a previous publication from Maggie Liem, who used to work in our group. So these 8,325 proteins, they were abstracted. We found them in 1.4 million cardiac-related cardiovascular papers, and these proteins were referenced in these publications. So that's how we got those 8,325 proteins. So what we would like to do is we'd like to uncover specific patterns of these 8,325 proteins across 6 main groups of cardiovascular diseases. These 6 groups are basically just textbook groups of heart diseases. And then we aim to gain new mechanistic insights and explore also new biomarker candidates in cardiovascular disease. And also what we would like to do is develop a natural language processing and machine learning platform so that for future purposes, we can analyze vast amounts of biomedical and clinical data. And then also maybe it's a daring statement, but what we also would like to do is that we can facilitate patient phenotyping and eventually even maybe do clinical decision making in the future. So this is just a concept figure. I think I've shown this many times before, but within our cardiovascular topics, we can focus on different types of text mining corpora that you see here on the left side. These are scientific articles in PubMed. That's what I'm doing in this study. But we also have been trying to focus on medical case reports. We're in process in doing that. And maybe although in the future, we could also think of other corpora like the electronic health records in the hospital. So through a text mining approach, what we would like to do is look for hidden patterns and relationship discoveries, and then this hopefully then can result in novel mechanistic insights that we could not see by the naked eye as a human being. So this is our workflow. We applied this workflow on 1.1 million publications, and they are from 1995 up till 2016. We used 8,325 proteins that were relevant in cardiovascular disease. And so the 6 heart diseases are basically, I'll highlight that later on a little bit more, but these are ischemic heart disease, the heart attacks, the cardiomyopathies and heart failures, CVA, that's more like strokes and brain hemorrhages, arrhythmias, the valve diseases, and congenital heart disease. So those are just more 6 textbook groups from heart disease that we used. And then our corpus was filtered to only contain articles with these 6 cardiovascular disease groups based on their mesh terms. That's how we collected them. And subsequently, every protein was then scored according to an algorithm that was developed by the team in Illinois. And we call it the OLAP algorithm. That stands for Context-Aware Semantic Online Analytical Processing. And then all these proteins would be ranked according to their integrity. Integrity highlights the meaningfulness of a protein within that corpus compared to the other heart disease groups. And then we would also look for their distinctiveness. And distinctiveness refers to their uniqueness in that heart disease group. And then we also would look at their popularity, which is more the frequency, so how many times they're mentioned. And then the overall ranking score at the end would be defined as a geometric mean of the 3 above mentioned criteria. So, so here you see a table with the 6 heart diseases, as I mentioned before here. You can see that bolded. And then you see the total number of abstracts that is contained in every heart disease. But it is also the case that one abstract could overlap with more diseases. So what you see here is also the overlapping abstracts. So ischemic heart disease on the left here, IHD, had the highest count of abstracts with about 385,000 abstracts, followed by cardiomyopathy, followed by arrhythmia, and then you have arrhythmia here, which is also, I think it's also very high. And then the CVAs, congenital heart disease, and valve diseases. And from all these disease groups, ischemic heart disease had the highest numbers of abstract that overlapped with the other cardiovascular disease groups. So, and then you see the highest count between ischemic heart disease and cardiomyopathy is over here. And then followed by ischemic heart disease and arrhythmia over here. So these are the most overlapping diseases in terms of abstract that we analyzed. And so we can explain this by either the popularity of being mentioned with another heart disease. So ischemic heart disease is often mentioned together with either cardiomyopathy or either with arrhythmia. But you can also speculate that there's a possibility that ischemic heart disease is often coexistent with either cardiomyopathy or with an arrhythmia. And that makes totally sense from a medical point of view. So ischemic heart disease often leads to cardiomyopathies and heart failure, and then ischemic heart disease often leads to arrhythmia. So in that sense, you can also make an interpretation in that way. So what we see here is that this panel displays the number of proteins on the y-axis versus their OLAP score in the x-axis over here. And so this is also for each of the 6 cardiovascular disease groups that we did that, and they're all depicted in a different color. The majority of these 8,325 proteins scored 0 actually in all 6 diseases. And it's only a total of 3,175 proteins that had a score, at least in one heart disease. So, and that's included in this figure. And from the previously, so only a very small, tiny subset of these proteins scored high within this case OLAP algorithm. So in each cardiovascular disease group, only a small group scored high based on the popularity, based on the integrity, and based on the uniqueness for that disease. So overall, what you see here in the brown line, the highest numbers of proteins were displayed in ischemic heart disease. That's depicted in brown, and you see that it's basically always a little bit higher than the other groups. And then you see also cardiomyopathies in green, and the CVAs is the other. It's in blue. And oh yeah, so over here you can I made a magnification here to see how that really goes when you have very high scores with only a few proteins over there. So our next challenge was to figure out what these scores, these case OLAP scores, would mean, right? So all we had was a number, and we wanted to put that into perspective, and we wanted to figure out what cutoff values we should apply. So we selected a couple of proteins that were highly relevant, that we knew that they were highly relevant within a heart disease. So if you look on your right side here, troponin I, this is the gold standard biomarker in clinic to diagnose a heart attack. So they're very relevant in ischemic heart disease. And we know that. And not only that, they're actually pretty unique in ischemic heart disease, because in all the other diseases, they don't really use troponin I. And if we look at that, it scored 0.18 on the as a case OLAP score within ischemic heart disease. So that was about in this area, 0.18. If you look at another protein, renin is highly relevant in cardiomyopathies, because renin values always change when your cardiac function change. And then it's also relatively unique in heart failure and in cardiomyopathies. And we saw that it scored 0.2 in cardiomyopathy. So that they're unique. So we thought like, hey, maybe in this area, these proteins have a high score. And we thought, oh, maybe let's see how it looks at 0.15 and higher. And they may be unique within a certain heart disease. If you look in this panel here, you see that CRP, this is a very popular protein in clinic. It's an acute phase protein. So with every acute phase in a disease, CRP usually goes up. And then we saw that CRP scored very high in ischemic heart disease, 0.2, because of course, a heart attack is an acute phase. But also if you look at other diseases like cardiomyopathies of CVA, then that scored 0.08. So then I thought, hey, maybe this area, this may suggest that this area may involve proteins that are still relevant, but more at least in at least 2 diseases. So then we took a look at interleukin-6, which is an inflammatory protein, and it also had the same thing. It overlapped in ischemic heart disease, CVA, and cardiomyopathy. So then in the next analysis, we thought like, okay, let's take a look at these cutoff values. So we displayed the number of proteins that were shared between the disease groups in different score categories here. And what we saw there was that if we implement a cutoff of a case OLAP score value, that this would highlight the uniqueness of the protein between the 6 groups of heart disease. So if you look at your left panel here, the left circular layout, This would be a score higher than 0.05, but lower than 0.1. And then you see that there's still a substantial number of proteins that are shared among the disease groups. And they are so basically not unique to one disease. If you look at the middle circular layout, then the number of shared proteins were much lower. And still you see that ischemic heart disease and cardiomyopathies, they still share the highest number of proteins that are shared, right? But if you look at the right circular layout, that is that cutoff score of 0.15, then you see that there's basically almost no shared proteins, very little, only a couple, like 3 or 4. And this implies that with this cutoff value of 0.15, the proteins that are scored there are unique to only one heart disease. So what we do here is we also made a heat map of all the scored proteins over the 6 heart diseases that you see here. Then you see certain protein clusters with a high score. And then we did a principal component analysis of these case OLAP scores within the 6 heart diseases over all proteins. And then you see that CVA, the strokes and the brain hemorrhages, cardiomyopathies and heart failure, and ischemic heart diseases are a little bit isolated, while congenital heart disease, valve disease, and arrhythmias are more clustered. And that would make sense because often a congenital heart disease would lead to a valve disease and vice versa, and they often are involved with arrhythmia. So this was a nice analysis showing that, you know, it's an interesting way of looking at these 6 heart diseases based on case OLAP scores in 1.1 million manuscripts. A human being could never be able, you know, could never do this in a short time. Then what we did here is on the left side, so now I look only at the score of case OLAP score proteins higher than 0.15. We knew that these proteins were unique to only one heart disease. So what you see here is on the left side in the heat map, you see indeed that there's really protein clusters that are unique to one heart disease. I highlighted here with a protein section that are that may be overlapping more than one heart disease. If you look at your right side, we did a cross correlation analysis on all the proteins with a case OLAP score higher than 0.15, and we indeed identified 6 main clusters that were unique for each cardiovascular disease group. So the largest protein cluster, cluster 1 here, this square, displayed 85 proteins that were unique to ischemic heart disease. And then we follow cluster 2, 43 proteins unique to cardiomyopathy. Cluster 3, 38 proteins unique to CVAs. And then cluster 4, 26 proteins unique to arrhythmias. Cluster 5 is 21 proteins. They were unique to congenital heart disease. And then the last is cluster 6 with 14 proteins in valve diseases. So again, I want to reiterate that we're talking about this area of proteins with a score higher than 0.15. We made also a table with the top 5, so basically top five single proteins with their highest scores within every heart disease, and that's depicted in this table. You see that for CVA, for instance, the most proteins were all in neurotransmitting. For ischemic heart disease, many of them were in lipid metabolism, and in the cardiomyopathies, we saw a lot of contractile proteins. Arrhythmias were all gap junction. Calmodulin is for calcium dynamics, so they all refer to action potential and indeed arrhythmias. And then the last 2, the proteins for valve diseases as well as congenital heart disease, are more connective tissue proteins. But again, these are just 5 single highest proteins that we put in a table. So now I want to talk about this area here. This area depicts a score between 0.1 and 0.15, and we knew from the previous data that these proteins may overlap more than one heart disease. So if you look here, from that area, we did another heat map, as you see on the left side. We did another principal component analysis. We basically see the same pattern in the PCA over the 6 heart disease. And then here we did, again, a cross-correlation analysis to identify 10 main protein clusters, as you see here. So we color them in red. So we did a cluster analysis using the Euclidean distance metric, and this illustrates the proteins that are clustered. And over here you see all these protein clusters. And they, for instance, cluster 1 are proteins that are overlapping in these 2 heart diseases. They overlap both in ischemic heart disease and cardiomyopathy. And as I mentioned before, these 2 diseases have the highest overlap. And indeed, you can see that here, that the number of proteins are the highest in these 2 diseases. But for instance, cluster 2, the number is much lower, but these are proteins that overlap 4 heart diseases, as you see here. So that's how we did that using the cross-correlation analysis. And then I made a table here. So this is actually the same table that you see here. The 10 clusters that I found here, I put them in a table, so cluster 1 up till cluster 10, and then say cluster 1 had 44 proteins. They overlap these 2 diseases, in this case ischemic heart disease and cardiomyopathies. We did an annotation using Reactome and UniProt, and then we found the key biological functions for these 2 diseases. And we found inflammation and the immune system, oxidative stress, and signal transduction. So the 44 proteins, they had these 3 key biological functions. And then here are just notable proteins like interleukins, tumor necrosis factor, STAT3 or superoxide dismutase, and etc. So we found those 10 clusters. And yeah, so that's, we did some more annotation to look at what's going on there. But it's interesting that in this case, with this score, in this score range, that you have proteins that are overlapping more heart diseases. So in summary, what we have now is we devised a natural language processing approach to annotate vast amounts of textual data from published manuscripts. So again, I don't think it would have been possible for human beings to get these kind of data without a 1.1 million abstracts. So a combination of phrase mining algorithms and large-scale network embedding technique is effective to recognize patterns and to extract relevant information, and providing novel biomedical insight regarding relationships in 8,325 proteins over 6 groups of heart diseases. And this natural language processing platform may also be suitable to mine vast amounts of accumulated patient information in clinical case reports, as well as in electronic health records. So I think, yeah, thank you for your attention.
Do we have any questions?
This is Andrew here. Very cool. I had one thought that may or may not be useful. I wonder if you could expand your text mining to include the search for variants, specific genetic variants, or mentions of genetic variants or protein variants. Because if so, then that could actually be a nice data set to import into ClinVar in terms of, because I bet a lot of those aren't in ClinVar currently.
Yeah, I think that's certainly possible, Andrew. Although that would, for variants, I think that would include a whole new realm into this equation. So I don't know what means we should take to look for variants. I mean, we took a look, I included 6 groups of heart diseases. And if you also want to take a look at the variants, I don't know what other source we could use to address that. I don't know if you have any advice on that.
Well, I was just thinking the variants, you know, it's sort of a high precision, low recall way of doing it, but you would simply look for RS digit for genetic variants. And then, you know, there are some, you know, similar types of just general patterns that people use to refer to protein variants too. So it wouldn't be as, I don't think, as complex as sort of the concept recognition that you do for the genes and diseases. But that's a wild speculation.
Okay. So what we were thinking about right now is to look at protein interactions. So with the data that we have now, we were wondering why these some of these proteins are more relevant, are unique to one disease, and one of them are a couple of them are more relevant in more than one disease, in 2 or more. And we would like to know whether this could be explained by protein interactions with each other, and maybe the variance would be an extra thing to consider.
Yeah, that sounds perfect, and that sounds actually more applicable to sort of a hypothesis generation type of activity. I guess the variance piece to me is more of like an open data, structuring data angle, where if, you know, you could relatively comprehensively claim that you'd, you know, categorized all variants associated with these 6 heart diseases and deposited them into ClinVar, that I could imagine being of great utility to all sorts of people. But anyway, I think, yeah, what you said is spot on.